I right, Mason? Science class, Matthew. It's okay to say. All right. We talked a lot about natural selection and survival of the fittest, weak traits versus strong traits, and, relative to whatever your environment is. And today we're going to look at artificial selection. But before we get into that, on 464, we're going to come back to this, but I'm going to hit on it while we're at it in the notes. You can see here the oldest fossils when they are formed, get buried under sediment and they have to be compressed over time. I'm going to bring some fossils from home and let y'all pick through them because I got way too many and they're sitting in basically some laundry baskets out by my garden, and my wife's telling me to get rid of them. So I'm going to bring some fossils. Y'all can look through them. We'll talk about them a little bit. If y'all want to take some of them home, you can. I always like to go fossil hunt. It's kind of like gambling. You just can't wait to see what you get around the next corner there. Am I right, Walker? So, also, let's flip over to, I don't know about the bonsai part, but 466. We talked a lot about Darwin. You can see his trip around the world there. They've got it outlined. One thing we noted yesterday is it took him about 23 years after his famous trip before he released Origin of the Species. You see a good picture of him right here. Who does that resemble? Nathan? Yeah, that's good. Nathan, you think you passed for Darwin?
I don't think so.
Okay, so, and then his finches that he observed on 467 and on 468. Like I said, we're going to get into making some cladograms, analyzing some cladograms. You'll have to do this on the AP exam for sure. You can see one here. Sometimes you see it referred to as a cladogram or phylogenetic tree. Basically, it's just a family tree showing an extended history of where organisms came from and how they got to where they are. And then we skip over to 469. So in 469, we actually catch up in the notes here to artificial selection. Artificial selection, read A for us, Matthew.
This is where man selects what traits are desirable, beneficial in a species. Plants, which ones make the best or most fruit or are the most appealing in the yard or garden. Domestic animals, which ones are the most valuable in terms of food or other characteristics.
Interesting. So artificial selection is where people are selecting traits that get passed on. Now, what are some good examples that we see of artificial selection? Breeds of what? Okay, dogs is probably the number one thing we think of just because we know about lots of different dog breeds. Now, we also know that dogs originated from wolves. And wolves were brought in, they were bred together, and to get certain traits. And over thousands of years, we have gotten quite a few varieties of dog breeds. See anywhere from your Chihuahuas to Great Danes. So we see a vast array of sizes, colors, different physical and mental characteristics of dog breeds. What else has been a product of artificial selection? Plants. So we may not normally think of it, but when you go to the store, there's probably rarely a produce item that you get that hasn't been artificially selected for. I would say probably other than maybe some tropical fruits, and a lot of those probably have been selected for as well. But other than some of the tropical fruits, I would say most things that you buy at the store have been altered significantly from the original plant. And a good example of that on 469 is wild mustard. So wild mustard, as it's shown here in the picture, has given rise through artificial selection to all of the following. The kohlrabi over here, broccoli, cabbage, Brussels sprouts, what is the other one? Kale. So all of those are related and products of breeding from those original wild mustard. Okay, which is pretty cool. You can get lots of different things. Why would you want to breed plants? More food, better nutritional value, better taste, longevity, how long will it stores or how long it lasts. Its ability to survive in different environments. Some species or some varieties of plants, produce that you get will survive in certain habitats much longer than others will. And so that can be beneficial, if you're moving from place to place and maybe environments are changing slightly. So plants are significantly different. One we can also relate to as plants would be corn. Corn back in the day when the natives got it was called what? Maize. And y'all see maize with the different colored kernels. When Native Americans or the indigenous people found corn, it would have looked something like, I don't know, probably like the size of a pen or a Sharpie, and it would have had just a few kernels just kind of scattered around. Not much to chew on. And so somebody in first period said, well, how do you go from something this size with a few kernels to something that's like this big and that big around with, hundreds of kernels? So how do you make that jump?
You slowly reproduce the better ones.
How do you reproduce them? Let's say you're a Native American, Mace, and you see this thing that has these kernels that you can eat, but you want to produce better varieties.
You want to make sure you plant the seeds of the ones that are have the most kernels.
Yeah, so you would go through, pick the ones that had the most kernels. You would take some of those kernels off of there. You would plant them, and you would plant a bunch of them. So they're going to cross-pollinate. Okay, so when you cross-pollinate, when you're undergoing sexual reproduction, you're going to increase the genetic diversity of your offspring through crossing over and random gamete selection and all that meiosis and all that stuff we talked about, and independent assortments. I can't think of the other thing. Anyway, so in doing so, you're going to wind up with some new corn that grows that has probably some different varieties or different amounts of corn kernels, and then you can pick the ones that are most productive there, get their seeds, and you just keep doing that. Maybe you find some that are different colors or different textures or that are sweeter, and so you plant those. Nowadays, we have quite a few variety of corn that you can pick from. You could probably narrow down corn to two different types, which would be what? White and yellow. That's not what I was thinking at all, but interesting. We have corn that's primarily used for
Starches.
eating. Us eating, we typically eat what type of corn?
Sweet corn.
Sweet corn. So there's lots of varieties of sweet corn, like peaches and cream, silver queen. I can't think of the others, but there's a bunch of different ones. And then you have other types of corn, I would just call it feed corn, that's grown for grain for cattle, and also used a lot in production of ethanol. And so you've got different varieties that are grown for different purposes, okay? The one like the sweet corn has a better, sweeter taste. You eat some corn, it's like really sweet and juicy and nice to eat. If you had to eat grain corn, you'd probably be like, man, that's disgusting. We grew some, what was it? It was like a purplish kernel variety, and there's a lot of varieties that are also used for flour production. And so, like basically, you would eat them and it wouldn't taste very good. It wouldn't be very sweet. But these are the ones that are used for grinding up, used for corn flour to make tortillas and other stuff that you use corn flour for. So, anyway, so there's lots of varieties, lots of interbreeding and crossing that's going on. Now we're in the age of genetics where you can do this at the molecular level, although I think, farmers have probably been doing a pretty good job all along in getting quite a good variety of different plants to produce. And now you see a lot of people who are going back to, they call them heirloom varieties of different produce, which are like older varieties. They contain a lot more diversity. There's a lot different types of taste. You don't wind up with just your cookie cutter store tomato. You wind up with ones that are sweet or that are a little bit sour or that, have a good, juicier texture, okay? So they taste quite a bit different than what you would get when you buy like your normal kind of just shiny, almost waxy tomato like at the store. So anyhow, lots of varieties through artificial selection. Domesticates, pretty much any domesticated animal has been done this way. Cows, pigs, chickens, turkeys, horses, goats, you name a domesticate, it's been bred. There's a breed with specific characteristics. Like chickens, we're raising chickens out here. We're raising chickens that are primarily for the purpose of production of eggs. So we have egg layers. The other type of chicken would be, meat chicken. So meaty birds that are grown out very quickly for eating. So those have been bred. And there's different breeds that have different characteristics that are, like we have Easter eggers and olive eggers that have colors, right? That have different colored eggs. So there's certain characteristics that people breed together that they like those characteristics. All right. Examples of artificial selection backfiring. Now, I'm going to go through A and C, and I'm not really sure about B. Somebody's going to have to look that up for extra credit because I don't know about as much about that one. But on A, Walker, read that one for us.
Insecticides creating insecticide-resistant bugs called superbugs.
Okay, so for those of you in here who've had apes, we talked quite a bit about this in the agricultural unit where if you are growing plants and you have a pest, so some type of like bug, and you spray for that bug, you're typically not going to get rid of every bug. Let's say, Ashlyn, that you spray your cornfield and you get rid of 97% of those bugs. 3% survive. So you got rid of most of them. Most of your corn crop is good. Well, what happens to those 3% that survived?
They're going to
They still like reproduce and have.
They're going to survive. They're going to reproduce. And their offspring are more likely to be resistant to that pesticide that you sprayed. And so maybe next year when you spray again, it only kills 90%. And then next year maybe 80. And then it keeps going down from there. So that's what we call pesticide resistance, where you're basically artificially selecting for the most fit individuals. You're killing off the weak ones, okay, because you want to get rid of them. They're hurting something you don't want or doing something you don't want them to do. And then you're leaving the most fit individuals to pass on their traits. Now, we also see this in the medical field with people, and this has a really bad impact on us. Read part C for us, Tay.
Mrsa is the antibiotic-resistant strain of Staphylococcus bacteria that cause staph infections.
Okay, so Mrsa, MRSA, is an antibiotic-resistant strain of staph. Now, when you guys see these big names in front of bacteria, don't freak out. Basically, like when you see Staphylococcus, coccus is just the shape of the bacteria, and the staph part is how it grows, like either in like small little colonies or rows or like how it's put together. So I know those are giant big words and they don't mean much to you, but it's really, they're just kind of defining how they look. So staph infections. I told you about my son had a staph infection like this, and basically about 10 or 15 years ago, somebody in the medical field is like, hey, we need to stop prescribing antibiotics. Why was that, Matthew? Look, you're deep in prayer over there. Why 10 or 15 years ago were doctors like, hey, stop prescribing antibiotics so much?
Because the diseases we used to evolve.
Okay, how do they evolve? Why, if you, let's say you went to the doctor and you had a really bad cold, why should a doctor not give you antibiotics?
What your body used to.
Walker.
Because the antibiotic would not only kill off the hostile bacteria, but the beneficial bacteria that's in it.
That's partially true, but Tay,
The bacteria, they're still.
colds are caused by
The virus,
viruses. Antibiotics kill bacteria. And so if you're using antibiotics willy-nilly all over the place, then you are going to be killing some bacteria. Let's shift gears. let's say you have a legitimate, that's one problem here, but it's going to lead into this other one. Let's say that you have a legitimate bacterial infection and a doctor, like an ear infection, and obviously it's bacterial and it's oozing everywhere and making a mess. And let's say the doctor prescribes you some antibiotics, Sarah. How does the doctor tell you to take those? What's the one thing they tell you? Mariah?
Take all of them.
Take them till they're what?
Gone.
Gone, or they give you a specific time period, usually 10 or 14 days. And so why do they do that, Nathan?
So whenever you don't take them for a few days and then stop, and then what's left over becomes resistant, and then
Yeah, let's say that you take them for 3 days and you start feeling better and you stop taking them. Well, you've killed probably a lot of bacteria and you started to feel better. But you maybe have left a few of the strongest, most fit individuals behind and haven't killed them all. And in doing so, then those bacteria start to replicate. Maybe you drink something and put it down and somebody else drinks it and they get sick, and you basically passed on a more fit variety of bacteria. And then in doing so, if enough people do that over and over, you're going to wind up with some super bugs that are antibiotic resistant and that we will not be able to fight off. Now, most antibiotics are made from what type of organisms? What antibiotics kill? What are antibiotics made from? Nope. Amoxicillin, penicillin, blah, blah, blah. All those are derived from. Fungi, good, yes. So remember, in nature, fungi and bacteria are constantly fighting wars with each other, and they're constantly trying to kill one another. Now, I think somebody brought this up earlier, maybe it was Walker, when we said if you take antibiotics and you don't need them, then it can kill some beneficial bacteria. Did you take that? So, for instance, let's say you take some, and even if you need antibiotics and you take them, it can still kill some beneficial bacteria, like in your stomach. A lot of times you take antibiotics, you get sick at your stomach because you're killing a lot of your natural, like, flora and your fauna in your body. And so it can also cause you to have a yeast infection, right? So, for instance, in babies a lot of times, when they are given antibiotics, they will develop thrush, which is a yeast infection in the mouth, and it has these little white patchy areas. I don't know if y'all have heard of this. And it's because you kill all the good bacteria, you leave a place for fungi to kind of take over. And so there's this constant battle always going on. And unfortunately, we have a limited supply of effective antibiotics. There's only a certain amount of ones that you can try if, like, it's not working against bacteria. And the problem is we're finding out that some of these antibiotic resistant bacteria are becoming more and more prevalent, and we may get to the point one day where we have outbreaks of, like, tuberculosis again, where we don't have anything to fight them off, and that would be a problem. All right, next page. Potential harm worldwide. You could see how this would play into our ability to survive if you have these outbreaks. Now, I'm going to give you another instance, and you apes people are going to know about this one. Let's say, Sarah, you're running a feedlot. You've got 100,000 cows you're feeding. You're fixing to carry them to slaughter. What's the one thing that you don't want to have happen with that $200 million worth of cattle? You got them all in a big feedlots together, you're feeding them out, they're all standing around eating, pooping, standing around in it. What's your biggest nightmare?
Them all dying.
Okay. What might kill them all? What's most likely to kill a bunch of them? What? Did you say diabetes? Disease. Yeah, what if you have some type of disease, pathogen outbreak, and they're all standing around together, they're drinking out of the same trough, they're eating the same food, and that spreads, quickly throughout the population, and, like, you have a massive die-off. That's millions and millions of dollars. So, Sarah, what do you do to prevent that massive die-off? You are constantly... Miranda?
Picking their gelatin and separating them out.
And what else are you doing? Yes,
Packing the food with antibiotics.
packing them full of antibiotics. The number one antibiotic use in the U.S. is feeding animals. So when you have lots of animals in a small space and you're trying to grow them very cheaply and inexpensively and as quickly as possible, disease can spread very quickly. And so if you don't feed them lots of antibiotics, you're bound to have some outbreaks of diseases. And eventually the worry is, and probably will happen at some time in your life, where there's some antibiotic resistant thing that starts killing a bunch of them and lots of them die off. And then where will all our hamburgers come from? All right, 473 in your book. Let's flip over.
Is that why on some packages they'll say like in chicken like 0 antibiotics?
Yeah, and so a lot of people were worried about the side effects of those excessive antibiotics, and so a lot of times on chicken packaging you will see no antibiotics used in the growing of these animals. I don't know if there's a significant difference, but I think they cut out antibiotic usage a while back for chickens. But in a lot of cattle, and I think probably swine too, you still see that. All right, at the top of 473, we got an interesting picture here. All right, tell us about homologous structures, Miranda.
Homologous means the same. Structures, examples include skeletal structure, limb structure, or cephalization. Remember, Darwin wrote about these in his book. Vestigial organs are organs that appear to have been needed in the past but are slowly disappearing.
Okay, so homologous structures. Homo means the same. So in this picture here, you see a human, a cat, a whale, and a bat. And they all have similar structures. Now, one thing that you need to it's really important is homologous structures indicate a common ancestry. So it indicates that there's some relatedness between things. So humans, cats, whales, bats, we all have some genetic relatedness that helps these forearms to develop. The humerus, now they may all be a little bit different shape, a little bit different size, but they're arising from the same embryological tissues, okay? And so like the phalanges on a bat are very similar to our phalanges, or a cat's, or a whale's even, if you're looking at like flippers. So these are things that are similar in nature. The same thing with the humerus. All of these are giving rise from similar tissues early on. A little bit different size and shape depending on what the organism is, but they are coming from the same tissue. So that means these things are all indicate that we are related in some way. Now, another thing that indicates relatedness is embryological homologies. So those are two big words. Those two big words just mean what though?
Embryos are the same.
Similarities between embryos. Good. So read part B for us, Ashlyn.
Embryological
Homologies.
are seen as common stages of development that embryos go through. Darwin wrote about these in his book to help show support for common ancestry.
Okay, so at the bottom of page 473, we have a good picture here. And we have a chicken and I'm going to see if I can cover it up here on the people at home. Oh. So we have a chicken on one side and we have a human on another. Just by looking at them, it's kind of hard to tell which one's which, right? They look pretty much the same. And I would venture to say if you guys saw most fish, chickens, reptiles, salamanders, amphibians, they would look very similar. I may actually give y'all a test later on in the week just for fun, for giggles, and see if you can pick out the embryo, like see if you can match it up. And the point being that pretty much all vertebrate embryos are very similar growing up. We all have these post-anal tails. You might got a tail still? You don't have to admit it if you do. We all have a tail bone though, so we all lost that tail at some point during development. We talked about apoptosis involved in that process. I also told you about my aunt who had like 3 extra inches on her tail, had to get chopped off. So, no offense, aunt. And then the pharyngeal slit arches right there. Those arches or slits that they're sometimes referred to as, those in fish develop into gills. Now, we have those early on and those go away, they heal up. But sometimes you see evidence of that left over. Like my son has a little in his ear, like right here, there's a little slit you can see in the skin. And so that's probably one of those pharyngeal slits that didn't heal up all the way. At least that's what my doctor for a mother-in-law says. And let's see. So basically, those are indications of a common ancestry among individuals because they develop in a similar way. Okay? Part C, Matthew.
Molecular homologies refers to DNA nucleotide sequences being exact in order and function. Darwin could not write about these as they had not yet been discovered yet.
All right, molecular homologies, similarities at the molecular level, so at the DNA level. So we have similarities in DNA with pretty much every organism on the planet, somewhat. I mean, like we talked about during, was it chemiosmosis? You remember that enzyme cytochrome C that's involved in pumping protons? Like that's found in every organism on the planet, that enzyme. So there's a genetic code for that. Obviously, if you switch gears to like us and apes, like a chimpanzee, I think it's like 98 point something % of our DNA is the same. So basically, the closer you are related, the more DNA you're going to have in common. The farther apart you are, the less you're going to have in common. But we all have certain things in common. Like I said, we're all using the same 20 amino acids, every organism on Earth, to produce the proteins that we produce. So we're using these similar things. We use a similar genetic code to do so. Okay? So when we start making family trees and things start branching off, you kind of have an idea of things are related. Just when they branch off, the farther it is down the tree or farther it is away means it's less related. All right, and then biogeography. So if you flip over to the next page, what is biogeography, Katie?
How, like, an organism, like, evolves to their environment, like, in different areas.
Yeah, so biogeography is where organisms are where they're located, and that's going to play into how they evolve because of the environment. Now, if you look at this picture on the, what number is that? 474. This is an example of basically a phylogenetic tree, which is kind of an evolutionary history. Some people refer to them like a tree of life. This only includes like a small little snippet here. And then you see little, what do they call them? Shared characters here. Things that are shared among organisms past that particular point. So like this one right here is digit-bearing limbs. So after this point right here, everything has a digit-bearing limb. Or this one, the amnion. Everything has an amniotic sac or an amniotic egg. So you're going to be constructing these. A lot of times they'll give you a blank one of these on the AP exam, and then they'll give you these shared characters, and they'll give you some different choices, and you have to fill in where everything goes in terms of its relatedness and what characteristics that they have. Okay? So you're going to see lots of those. Now, endemic. Endemic species are organisms that are found where?
Only in one place.
So they're only found in one specific place. I'll give you an example. In Hawaii, about 25% of the species there are only found in Hawaii. So if they're only found there, where did they come from? Did they magically appear in Hawaii?
No.
No. Well, then how did they come to be?
They had to have migrated from a nearby, major land structure.
Okay, some had to migrate there, and then the organisms had to.
Change the environment.
So some of those organisms, yes, evolved and changed over the course of time. And I imagine, like Walker said, if they migrated there somehow, if you were to look at these locations around the Pacific where they may have come from, you would probably find some similar relatives of those organisms. Similar plant species, or I don't think there's any snakes there. Similar lizards and maybe other insects that you find there. But probably a little bit different, okay? Different enough where they're their own species. Now, we're all talking about how things are related and how these traits are passed on. Let's talk about how another aspect of evolution is called convergent. Convergent means what? Now, this isn't that movie with Katniss, and the girl with the bow and arrows and all that stuff, and you know what I'm talking about. This is a different thing. What does convergent mean?
It means to like come together, sort of.
Come together right now. Yes, to come together. So, convergent evolution, I'll give you an example. If you look at the top of 475, we have our friend, the flying squirrel, okay? And we have a sugar glider. So, a flying squirrel, I've only seen one of these around here, and it's because it ran into a window and basically passed out for a while. I think it had a concussion. And it was at nighttime, and so that's the only time I've ever seen one. But we have flying squirrels around here. And we also have our friend the sugar glider, okay, from over in Australia. Now, if you look at them, physiologically, they look almost the same. What is the one main difference between those two organisms?
Location.
Well, they're found in different locations, but does anybody know the one main physiological difference? They are not in any way like related, like closely related. Sugar gliders are, they're closely related, more closely related to kangaroos than they are squirrels.
So they're marsupial.
They are marsupials. So even though they look the same, sugar gliders are marsupials and they evolved from other marsupials, and flying squirrels are placental mammals, meaning they have a placenta that attaches to the offspring and nourishes it longer. And so they are significantly different in their evolutionary history. Now, they wound up being about the same, and so that's why we call this convergent evolution. Things have converged or come together, or they're almost the same because of the environment that they grew up and developed in. You could think of this also in terms of, like, let's say you take penguins. Think penguins in the water, not penguins on land. Penguins, sharks, dolphins. What do they all have in common?
Flippers.
Okay, they have flippers. If you look at them, they have this like torpedo body shape. It's called fusiform, Fusiforme. Yeah, not E at the end, form. And so fusiform bodies are usually developed for organisms that are predators in the water to help you move quickly through the water. If you don't know anything about penguins, they move really fast through the water. They're very good fishers. And so obviously a penguin's a bird, a shark's a fish, a dolphin's a mammal. These things are not really closely related, but they all have very similar like fusiform body shapes. And that is because of this convergent evolution that's taking place where they're all in very similar environments, hunting very similar items, okay? Now, speaking of those structures, number 2, Mrsa, read that for us. Analogous structures.
Analogous structures have the same function, such as a bat wing, which has bones and muscles, and an insect wing, which doesn't have muscles and bones in it, but they both produce flight. Do not confuse homologous structures. Homologous indicates common ancestry, and analogous does not.
Okay, analogies. What's an analogy?
Just like a way of describing something.
It's a comparison between things that are similar, right? So when we talk about analogous structures like the sugar glider and the flying squirrel, they have analogous structures, but that does not indicate a common ancestry. So like a bat wing and an insect wing, those are both wings that help them fly, but that doesn't indicate that they didn't inherit that through some common ancestor, okay? So make that distinction. Homologous structures indicate a common ancestry. Analogous ones do not, okay? And then the last part of the notes, fossil evidence supports the theory of natural selection by displaying common structures between species. The fossil record is incomplete because of the nature of how fossils are made and the fact that some are still undiscovered in the dirt or at the bottom of some body of water. Now, we haven't talked much about fossils yet. We're going to look at quite a few of these that I have around the room. I got one over there of a fish that I got out in Wyoming. But fossils don't form very easily. Like if one of y'all died out in the woods somewhere, I mean, something would probably eat your bones and they'd chew on them and they'd break them up and some of them would get scattered about. And if there wasn't a lot of stuff around and things covered it up over time, you might get fossilized. But a lot of times they just kind of get broken and scattered and there's not much left. And so if you fall in next to a body of water and get covered up with sediment, and over time you might get fossilized, but it's kind of hit or miss. So that's why we have kind of this incomplete fossil record. It's just not, fossil formation isn't as easy as you might think. A lot of factors have to go into play to forming fossils. Okay, so any questions?